So we go to the next speaker, that is Murali Prakriya. I hope the pronunciation is okay. He's a professor of pharmacology at the Northwestern University of Chicago. And the title of the talk is The Molecular Anatomy of Store-Operated Orai Channel Gating. Please, Murali.
Great. Thank you very much, Luigo. And I would like to thank the Alberto and the other organizers, and of course the session chair for putting together really a fabulous, absolutely fabulous conference. I've been really enjoying the live speeches and the recordings as well, and it's really a pleasure to see all these talks on the biophysics of nanopores and ion channel gating. So I'm going to go put my.
Orai, sorry, can you make full screen, please?
Yes, sorry.
Okay, okay, it's okay
so
now.
Let me also start up the pointer. Can you see the arrow? Can you see my arrow?
No, try to move it.
Let's see. I'm going to try again. Let's see.
No, I don't see the arrow.
Okay. Sorry, let me try that again. It was screen. Oh, here it is. Laser pointer.
Okay. Now I see it.
Excellent. Thank you. Sorry for that. So yeah, I'm going to be talking about basically the activation mechanism of the store-operated Orai channels. So I'm in the pharmacology department at Northwestern, which is in this jungle of buildings that's located right here in this cluster. If anybody has been to Chicago or if planning to come to Chicago once the pandemic is over, please come by and say hello, and we'd love to have coffee with you. So my laboratory works on the molecular physiology of these Orai channels. Historically, they were referred to as crack channels for calcium release activated calcium channels. So what are these store-operated crack channels? They are basically highly calcium selective channels in the plasma membrane of probably most of our cells that are activated by stimuli that deplete ER calcium stores. Now, physiologically, stimuli that deplete ER calcium stores can be things like receptor tyrosine kinases or G protein coupled receptors that result in the production of the diffusible messenger IP3. And when that activates IP3 receptors in the ER membrane and discharges the ER calcium store, it causes a lowering of the ER calcium concentration. And that lowering of the ER calcium concentration then activates these channels, these calcium channels in the plasma membrane, which then produces calcium influx into the cell. And that calcium influx not only refills the endoplasmic reticulum store, but it also directly activates a variety of effector functions that can range from gene expression to regulation of migration, synaptic plasticity, exocytosis, and so forth. Now, the molecular components of these channels were identified in 2006. So up until then, the channel was known as the crack channel. And in 2006, several groups discovered that Orai, the family of proteins called Orai, of which Orai1 is the best described member, forms the calcium selective pore or the calcium channel. And it is activated by the ER calcium sensing proteins, of which STIM1 is the best described member. So there's two STIMs, STIM1 and STIM2, and there are three Orais in our bodies, Orai1, Orai2, and Orai3. But most of the historical work and today's talk is going to focus on the best studied member, which is Orai1. Okay, so what are the physiological, why are they important for human health? So in fact, I should point out that Orai was discovered as a part of a linkage analysis screen in a family of human patients that suffer from a severe immunodeficiency. So this immunodeficiency affected both T cells and B cells in the immune system that resulted in a very severe combined immunodeficiency that made these patients, these are children typically, they would be susceptible to opportunistic bacterial and viral infections, and that frequently, if not always, actually for ORI1, results in lethality within the first year of life. They don't survive beyond 6 months to a year. In addition to that, patients who get a bone marrow transplant will have other serious problems like muscle weakness, autoimmunity, and then they have defects in the formation of skin and nails that is referred to as ectodermal dysplasia. So that told us that these channels are actually quite important for human health and especially for activation of the immune system. Now, since Orai was discovered, many gain-of-function mutations that cause constitutive activation of the channel have also been identified, and these defects are much milder. The most prominent of these is a muscle weakness that arises from a tubular aggregate myopathy, where the fibers of the muscle aggregate together. And since then, many groups, including my own lab, we have made knockout mice of specific isoforms of the Orais as well as the STIMs. And so what we know about these, the role of these channels is that they regulate things like inflammation, cell proliferation, regulation of epithelial cell function in the lung, skeletal muscle contraction, and also they're expressed in the nervous system where they regulate synaptic plasticity and calcium signaling in astrocytes and microglia. So over the years, my lab has focused, after the discovery of ORI1 and STIM1, my lab focused on the activation mechanism of the Orai protein. So in other words, once STIM binds to the channel, the ER calcium sensor, how is the channel activated? And STIM binds, I should point out, to the C terminus, predominantly to the C terminus of the cytoplasmic C terminus of the Orai protein. So the questions, this big question of how is Orai gated can be broken down into its component subparts. So we wanted to know where in the ion conduction pathway is the channel gate, what is the nature of the gating mechanism, and more broadly, what are the conformational changes in the pore that underlie channel activation. So this work was predominantly done in my lab by 3 individuals, a former postdoctoral fellow, Beth McNally, who's in Maryland now, a senior staff member in my lab, Megumi Yamashita, and a former graduate student, MD PhD student, Priscilla Young. So the first thing that when Orai was discovered in 2006, it was when we looked at the sequence, it had very little, if any, homology to other ion channel proteins. So we couldn't just look at the sequence and say this is where the pore is, or that might be the gate, or what have you. So in order to understand which part of the protein forms the ion conduction pathway, we used a cysteine accessibility analysis where you use water-soluble thiol reagents to look at and in cysteine mutants of the Orai protein to see which residues underlie the ion conduction pathway. And so that information, those set of experiments told us that it was predominantly TM1, transmembrane domain 1, that forms the ion conduction pathway. And we also identified several residues in this long helix of about 24 angstroms that were the pore flanking residues. So for example, D110 in the outer mouth of the channel, followed by the selectivity filter, which is located at 106, and then V102, G98, L95, and R91. So once we knew that TM1 forms the ion conduction pathway and we had identified the residues, before that I should point out that a few years later, after we published our work, Steve Long's group at Memorial Sloan Kettering published the crystal structure of the closed Drosophila Orai channel. The Drosophila Orai channel is very similar to the human Orai channel in its functional properties. It also forms a highly calcium selective store operated current. And in this beautiful structure, you can see that the channel is actually a hexamer, which was a surprise to many of us at the time. We were expecting it to be a tetramer. And the ion conduction pathway is formed by the transmembrane domain 1, and these carboxylates here cradle the calcium ion at the glutamate selectivity filter. And we are very gratified to see that, in fact, the pore lining residues that they had identified from the crystal structure, E106, V102, L95, and R91, were also the same ones that we had identified from cysteine accessibility analysis. The only point of significant difference was that whereas the crystal structure of the closed channels showed this phenylalanine at 99 as the pore lining residue, we did not get any reactivity to this phenylalanine. Instead, we found that it was G98, the neighboring residue, that was reacting to thiol reagents in the STIM1 activated open channel. And I'll get back to this important difference in a few slides when I talk about gating. So the next step, the next question that we wanted to ask was, where in the ion conduction pathway is the ion channel gate located, and what is the nature of the gating mechanism? What is the nature of the gate itself? So for this, we again used cysteine accessibility analysis, but instead of just only looking at the open channel, we looked at the accessibility differences between closed and open channels. And the idea is that if there is a closed channel gate located somewhere along the ion conduction pathway, that closed gate will occlude or prevent the access of thiol reagents below the point of closure in the closed channel, but not in the open channel. So that told us, those sets of experiments told us that the ion channel gate is located somewhere between D110, that's located in the outer mouth of the channel, and this glycine residue that's located somewhere halfway along the middle of the transmembrane segment. And the only two amino acids that are located between these two loci are E106 and V102. E106, as I mentioned earlier, is the glutamate selectivity filter, and as such, there is no data that it's involved in gating, and so that left us with this valine at position 1 or 2 as a likely candidate for the ion channel gate. Now we figured that if this valine has an important role in gating, then if you mutate this valine, then it should destabilize Orai gating. And so, in fact, consistent with that, what we found is that mutating the valine to amino acids that were less hydrophobic resulted in constitutive activation of the channel. So normally, Orai is a two-component system. You need not only Orai, but you also need to need STIM1. You need to express both of these proteins in order to reconstitute the endogenous current. If you only express Orai1, you get nothing. Whereas in this V102C mutant, the cysteine mutant, this channel was open all the time. It didn't need STIM1 for it to be open. And what we found by replacing the native valine with a series of hydrophobic amino acids is that it causes mutations that reduce the strong hydrophobicity of this valine, cause constitutive activation. So alanine, glycine, cysteine, threonine, glutamine, they were all constitutively active. So we came to the conclusion that this valine must regulate ion conduction by hydrophobic occlusion of ion flux. Valine is an extremely hydrophobic, oily amino acid, and so it was reasonable to think that it might present a hydrophobic and free energy barrier to the occupancy of water and ions in the ion conduction pathway. Now, one thing I should point out is that when we looked at the ion selectivity of these and other mutants, there was something interesting that came out, and that was that the native Orai channel, the wild type channel, is one of the most calcium selective channels that's known in our body. In fact, it might be the most calcium selective ion channel, more selective than voltage gated L-type calcium channels. And so that's reflected in the very, very positive reversal potential in the current voltage relationship. This channel is also very weakly permeable to large monovalent cations like cesium. The valine mutant, in the cysteine mutant of the valine, was much, much less calcium selective. In fact, it was mostly non-selective, and the cesium to sodium permeability was about equal. But when we took the mutant and put STIM back into the system, so when we activated it now, it can be further activated with STIM1, then the properties of this current looked indistinguishable from the wild type channel, and everything was normalized. Ion selectivity was normalized, and other features of the pore properties were also normalized. In fact, when we looked more closely at the changes in the reversal potential as the channel gets activated, we found that with increasing amounts of STIM1 ligand on the channel, not only was the current size increasing, but the ion selectivity was also shifting towards more calcium selective channels. So this is an interesting case where ion selectivity and channel gating are very intimately coupled in Orai channels. I think it's a very clear example of that. Okay, so back to the question of how does this valine work potentially, if this is a part of the gate, how would it actually work in opening and closing the pore? So if you look at a cartoon, a top-down view of each of the subunits, and you look at which pore, where are the pore lining residues, the valine is actually located right in the middle of this pore exposed surface. Valine by all accounts, by any measure, it's not a very interesting amino acid. It's short and stubby and just has these two nonpolar side chains that protrude into the lumen. And what we found is that it's exposed right in the middle of this pore exposed surface for each subunit. So what that told us is that the conformational change that must remove this valine, if it's a part of the gate, must be pretty substantial. It would have to be something significant to conceal it away from the ion conduction pathway. But as a first step to look to see, is it in fact displaced away from the ion conduction pathway, Megumi put a cysteine over here and looked at the accessibility of the cysteine to the thiol reagent cadmium. Cadmium can be coordinated by cysteines and it would block ion conduction. And what we found is that whether the channel was bound to STIM1, which is its natural ligand, or unbound to STIM1, there was basically no change in the accessibility. It was equally accessible both in what would be closed and open channels. And the same result was, we were puzzled by this, so we tested other thiol reagents like silver, got exactly the same result. And so what this told us is that the exposure of valine to the pore is not altered significantly or substantially by STIM1 binding. So that was very puzzling to us. And so we then considered the likelihood that maybe, in fact, this is not the dynamic part of the gate and that the gate must lie somewhere else in the ion conduction pathway. And so that then led us back to the crystal structure, back to the drawing board, where it had shown that the crystal structure had shown that this large amino acid phenylalanine was in fact the pore lining residues, not the valine, not the glycine next to it that we found by cysteine accessibility analysis. Now, if you look at the positions of this phenylalanine and the neighboring residue 98, they're located at the very edge of this pore exposed surface. They're neighboring residues 100 degrees apart. And so, it might seem reasonable that it might not take much to conceal F99 while bringing in G98 into the pore. So to directly look at this question, we looked at the accessibility of F99 by putting a cysteine there, as well as G98 by putting a cysteine there, and putting the V102A mutation to constitutively open the channel, and looking at their accessibility in the absence or in the presence of STIM1, which is its natural ligand. And what we found is that whereas the accessibility for F99 dramatically decreases, it's readily accessible in the STIM1 free state, but when you gate a channel with STIM1, its accessibility dramatically drops off. By contrast, exactly the opposite pattern is seen at G98. Here, G98C is not accessible in the STIM1 free state at all, but its accessibility dramatically increases following STIM1 binding. And when we looked at the other pore lining residues like V102, as I mentioned already, L95, etc., we didn't find any other changes in the accessibility of those residues. So in essence, what was happening is that the pore lining residues were changing from the bar that is shown in yellow over here, which includes 99 in the closed state of the channel, but excludes 98, to the bar that is shown in orange, that now excludes 99, but includes 98 in the open channel. And we figured that the simplest conformational change that could potentially explain this type of displacement is a modest, a small 15 to 20 degree helix rotation that would conceal F99 away from the ion conduction pathway while bringing G98 into play. So the next question was, well, if F99 is in fact the dynamic part of the channel gate, then again, mutations at F99 should destabilize ion channel gating. And so consistent with that, when we decrease the hydrophobicity, phenylalanine, like valine, the one turn below it, is extremely oily and hydrophobic. So if you decrease the oiliness or hydrophobicity of this phenylalanine, then you get constitutively active channels. By contrast, if you increase the hydrophobicity by substituting valine, isoleucine, or leucine, in fact, these channels are not only not open in the absence of STIM, they're even harder to open in the presence of STIM1. Now, to more quantitatively address what exactly is happening in terms of the free energy for water and ion occupancy, we collaborated with Regis Pomes and his graduate student Chris Singh in Toronto, and they performed molecular dynamic simulations on a variety of different mutants. And what we found from their analysis is that, in fact, if you look at the hydrophobic part of the ion channel, substitutions like V102A or F99Y, which strongly activate the channel constitutively, result in significant increase in water occupancy in the hydrophobic stretch of the pore, whereas the valine mutant actually decreases water occupancy to a modest extent. Not only that, interestingly, we also found that these mutations, like especially F99Y and V102A, result in counterclockwise fluctuations, increased counterclockwise fluctuations away from the ion conduction pathway, which would cause the channel gate to get displaced away and increase the water occupancy and decrease the free energy barrier for ion conduction in the pore of the channel. So in other words, when an ion enters the ion conduction pathway, so this is now sort of the model, ion conduction pathway, you can see that in the resting state in the crystal structure with a barium ion that is stuck at the selectivity filter, with these phenylalanine sort of in a pore-facing orientation, there is very little likelihood that this is going to have sufficient opportunity to pass through the ion conduction pathway. When STIM1 binds to the channel, these phenylalanines, through an unknown mechanism, displace away from the ion conduction pathway, but the exposure of this valine that's located 1 ton above is not significantly altered because it's located right in the middle of the pore exposed surface. So the dynamic part of the barrier is regulated by the displacement of F99 in and out of the ion conduction pathway. Now, the next question that we wanted to address was, well, that's all well and good, but how is the signal actually transmitted from this distant C-terminus binding site to the ion channel gate that's located towards the extracellular part of the ion conduction pathway? What is the relay mechanism? Is it going through a direct interaction with the pore, cytoplasmic extension of the pore, as some people had postulated, or is it going through some other pathway? So to look at that, Priscilla Young, a graduate student in the lab, took advantage of pre-existing cysteine mutants that we had generated in the lab when we were looking at cysteine accessibility analysis. And she also generated a variety of other cysteine mutations in all 4 transmembrane segments. This is a really deep mutagenesis scan in order to see which of these mutants or which of these loci are important for STIM1 activation of the channel. And much to a surprise, what we found is that there were a very large number of mutations that are highlighted in these yellow dots that were all constitutively active, even in the absence of STIM1. And the interesting thing is that so if you look to see where these mutations are located, some of them are located about 30 angstroms away from the ion conduction pathway, all the way in transmembrane domain 4. So somehow mutating these residues in transmembrane domain 4 very far away from the ion conduction pathway seems to remotely switch the gate into the active configuration. The ion conduction pore properties of these channels were quite variable. Many of them were actually very, very calcium selective, like this mutation at H134. But some others were actually not so calcium selective, like this mutation at E190, but they did become calcium selective when STIM1, the ligand, bound to the channel. When we plotted the amount of current that was already active at baseline on the x-axis versus the amount of current that could be further activated when the channel is bound to its ligand, STIM1, we got this sort of relationship, sort of an inverse relationship, so that Excuse me. So that the mutations that are already very active, like this H134 mutant at baseline, could not be significantly gated further by STIM1 binding, whereas others that are only weakly active at baseline could be strongly activated further by STIM1 binding. So the one outlier that really stands out here is the mutant at H134. So H134 is located behind the pore. So this is the pore helix. And when Priscilla did a further analysis of what's going on with this locus, what she found is that if she decreased the size of this large histidine, then the channel became constitutively open. So we think that this histidine, through a hydrogen bond interaction with a backbone carbonyl, with the backbone nitrogen, is working as a steric brake to flip the channel into the open state when it's released. And so here is a molecular dynamic simulation movie of both the wild type channel and this H134 mutant that's located right there. And you can see that in the wild type channel, this phenylalanine spends most of its time oriented in a pore facing configuration, so that the pore is only modestly occupied by water, these little squiggly molecules. Whereas in the H134C protein, the mutant channel, you can see that the F99 gate is strongly displaced away from the ion conduction pathway, and that increases pore hydration, resulting in constitutive activation of the channel even in the absence of STIM1. Now, Priscilla also did an extensive what's called contact dots analysis and combined that with hydrophobicity analysis, and that gave us patterns of domains, revealed some interesting domains that were otherwise not visible. And one of these domains that jumped out was this cluster of hydrophobic residues that were located in the very top of the TM1 helix towards the extracellular side of the channel, that were reaching out and making contacts with the neighboring layer of helices that surrounded the pore helix. So they were reaching out and making contact with these helices, and some of these were very interesting residues. They were all extremely strongly hydrophobic, and that's to be contrasted with, as you go further into the cytoplasm, you get these alternating stripes of hydrophobic and hydrophilic residues that seem to have unique roles in gating the channel open. But we focused our attention on this hydrophobic cluster, and very interestingly, when we looked at the contacts in the molecular dynamic simulations of one of these residues, which is a methionine that's located on TM1 itself, in the pore helix, but in the sort of a non-pore facing side. When we looked at this methionine, what's going on here? So what we found is that this methionine in the open channel was making extensive contacts with the channel gate itself. So the question, the real question from the previous work is, if F99 is a channel gate, what is the signal that's actually causing its displacement away from the ion conduction pathway? And in the wild type channel, which would be closed in the absence of STIM1, this methionine residue, which has sulfur, was predominantly interacting with its resting state partner, which is these are all Drosophila numbers, so this would be F187 in human Orai, and this is the F99 gate that I was talking about. So this was an exclusive interaction between the sulfur-containing methionine and another phenylalanine that's located in the next layer of helices. Whereas in the constitutively open H134C channel, you can see that there seems to be a switching mechanism where this methionine is now intimately interacting with the channel gate and spending considerable amount of time engaging in interactions. So this was very notable, and it caught our eye, and we wondered, well, is this part of the stimulus that promotes opening or displacement of the F99 gate? So in collaboration with Regis Bomis and Chris Singh again, this is a busy slide, but I would like to draw your attention to panel E, where what we did is we basically just looked at the this is a plot of the distance distribution between the sulfur atom of that M101 residue. These are again Drosophila numbers, so it would be M101 in human, and the channel gate, so the center of the phenylalanine ring. And what we found is that in the wild type channel, these distance distributions between the gate and the methionine is actually quite broad, and it spends really a lot of time at distances that are larger than this magic black line, which is it would be in a range of distances where there wouldn't be any meaningful interactions. Whereas in the open channels, there's a noticeable shift, so that these distances are now shifted towards much smaller values, which would indicate a close reaction. And so we were really curious about this, because these sulfur-aromatic interactions between methionines and other aromatic residues like phenylanilines have been frequently described in protein structures to stabilize protein-protein interactions or domain interactions, but then has never been described in ion channels, and in fact have not been previously described to have a role in gating. But because sulfur with its slight negative charge can potentially attract the phenylalanine, we wondered whether this interaction might be actually what's causing the F99 gate to displace away. So as an experimental test to ask, well, if you artificially promote an interaction between these two positions, between F99 and M101, what will happen to channel gating? So what Priscilla did is put cysteines at both loci, and then she put cadmium, which will form a metal ion bridge to bridge these two positions. And when she did that, she found a huge extent of activation of the channel, really several-fold increase in the amplitude of the current. And interestingly, if she increased the concentration of the permeant ion in the ion conduction pathway, you could actually significantly decrease this cadmium-induced gating effect of this double mutant, as if the cadmium that is large or this mediating this effect can actually feel the concentration of flowing ions in the pore, which would be consistent with a location that is close to the ion conduction pathway. Now, by contrast, if she puts cysteines between the methionine residue and the resting state partner at F187, and then again applied cadmium, she could induce very rapid channel closure, which would then revert the methionine back to its resting state and free up the gate to be in the open state, in the closed state, excuse me. So it would block the ion conduction pathway. So this is basically would be analogous to deactivation. And in this case, it was a lot less permeant to the concentration of ions in the ion conduction pathway, which would be consistent with the location that's further away from the ion conduction pathway. So as a test of whether this methionine, if it is important for gating, what would happen if you destabilized this particular position? And consistent with the idea that it's very important for opening of the gate, nearly all mutations that, in fact, all mutations that Priscilla tested, including hydrophobic amino acids like leucine and isoleucine, were unable to sustain the opening of the channel in response to STIM1, indicating that hydrophobicity by itself is not sufficient to promote the interaction between these two residues, but you need the sulfur atom in the methionine group to induce a sulfur-aromatic bridge to open the channel gate. And so we call it the gate-latch that promotes opening of the F99 gate. So we think that it has the optimal combination of length, hydrophobicity, and chemistry with its sulfur atom to act as a perfect latch for the hydrophobic gate. And in fact, if you sort of a preliminary analysis of the free energies of these interactions based on the probabilities of the distances, of the occupancy of the distances, told us that it contributes somewhere in the range of about 0.8 kilocals per mole, or about 5 kilocals per mole for the entire channel, which would be pretty significant, I would think. We don't know what the free energy is to activate the entire channel yet, but by comparison to the activation energies for voltage-gated potassium channels, we would think that this would be a meaningful free energy contribution. So in summary, Orai channels use a hydrophobic gating mechanism for controlling ion flow. The displacement of the gate is mediated by a sulfur-aromatic gate latch between a methionine residue and the neighboring helix and the channel gate itself that promotes opening of the pore. And overall, these results provide a nice framework, I think, for understanding the allosteric interactions between distant domains for Orai gating, which would be compatible with a gating signal that's transmitted through the transmembrane domains. And again, the work was done, the initial work was done by Beth McNally, and some of the later work that I described was done by Megumi Yamashita and graduate student Priscilla Young. And of course, we are grateful to our collaborators, Regis Permis and his graduate student Chris Ng in Toronto. They're really awesome, and it's a pleasure to interact with these guys. And I'll take any questions that you may have. Thank you. I'm sorry, I'm not.
I'm sorry, you didn't hear me. I was muted, so sorry.
No, yeah.
So I was thanking you for your very nice talk, and for the moment I don't see questions on the chat, but I have one question.
Sure.
I was very impressed from the change in selectivity of the channel when you let the channel to interact with STIM. So do you have an idea of the mechanism of this selectivity change? Maybe the water below the glutamate might change the selectivity of the ring of glutamates?
Exactly right. So we think that the rotation of the pore helix would be, because the gate is located only one or two turns, the valine and then the phenylalanine. They're very close in. They're only 2 turns away from the glutamate selectivity filter. So a local twisting or rotation of the TM1 helix, even if it's a modest one, would be presumably detected as a conformational change in the orientation of those carboxylates, and so that might account for the changes in the
but this rotation of the helix
does
involve also the glutamates? The glutamates rotate a little bit?
Absolutely. So I didn't show that in the molecular dynamics simulations, but there is if you look at the position of the C-alpha atom of the glutamate residues, there is a 10 to 15 degree, a little bit less. So the twisting of the helix appears to be most pronounced in the hydrophobic stretch, but there's a little bit of propagation up and down the hydrophobic stretch of the pore helix.
Thank you. There is a question from Alberto Giacomello. Could you please comment on the formation of the bubble in the hydrophobic gate? Is it stable?
So, yeah, I was watching this very interesting, very nice talk from Paolo yesterday morning. And I think he may be able to speak better on this particular question. But very nice work, Paulo. And so just as a way of background, Paulo was looking at the correlation between water occupancy and the fluctuation of the F99 gate in MD simulations. So all our data is actually a summary data. So we were not looking at the temporal correlation. And it appears that from Paulo's data that in fact a fluctuation of the F99 gate, and it might be very cooperative, so that you can imagine that because of the interactions with all these methionines, the twisting of these helices may all happen in a highly cooperative fashion. But at least the data from Paulo's slide seems to suggest that there's an abrupt increase in water in the hydrophobic stretch, which would be consistent with a sort of a stable, at least on the timescales of these simulations, stable displacement of the ion conduction gate in the active state. And one thing that I didn't mention is that these Orai channels, they have a very high open probability. So when they open sort of in a stepwise fashion. to POs of about 0.8. And that would suggest that the displacement of the gate, it's kept in the active configuration stably, which would be consistent with what Alberto is asking.
Thank you very much, Murali.